Okay, folks, this is just a trial run of a new setup I'm using here. What I'm using is I've got an iPad, it's a M3, it's not the newest one that's just released, it's the last year's one, an iPad M3, and it's a 13-inch version. I like the really big screen, it helps me a lot while I'm doing my private tuition. So what I'm going to do today is I'm just going to try and see how we get on talking you through the carbon cycle or the carbon fixation stage of photosynthesis, which is found in higher biology unit 3, right early on, and I think it's 3.2. Don't quote me on that off the top of my head. So, what do we need for this carbon fixation? I mean, there's two questions that you're going to ask. First of all, is it controlled by anything, right? So if you're asked in an essay, the first thing that you want to do is you want to put it's a series of enzyme controlled steps. Okay, it's a cycle. Things happen in the cycle at different stages, different changes take place, and enzymes don't tend to be far away, but we'll highlight the enzymes that are going to be most important for it. Okay, so as it's a cycle, right, if you bear with me, if you're following along and you're going to draw with me, okay, give it a wee bit let me go about a second or two ahead of you, because sometimes the shapes snap, because honestly if I did it freehand it would look like an absolute mess in a dog's dinner, but the shapes do tend to snap. Now, first of all, I'm going to draw a half decent sized circle, okay, bring it out a bit, and as it's a cycle, it's going to go continuously round and round and round, as long as photosynthesis is taking place, feeding it all the raw materials, I suppose, then it's going to take place. So, what happens is, for carbon fixation, there's always a good place to start and make the link here. The link I'll make is that if you're going to do the carbon fixation, always start with carbon dioxide, right? So right at the top, I'll change the colour, okay? We've got carbon dioxide, okay? And what's going to happen is it's going to feed into the cycle. No, sorry, I'm trying to curve that in. Nah, it's not working. There we go. That's better. Okay. Yeah, so we feed it into the cycle like so. Okay. And I'll just put an arrow sign to show it's going in the way. So, carbon dioxide is going to join this cycle, but within the cycle, there's already something in there that the carbon dioxide needs to meet. So what I'm going to do is I'm going to duplicate this square and put this over here, make it a little bit bigger. And what you get is you get the substance, it's RUBP. Okay, so the carbon dioxide is going to join RUBP, but because we're going from RUBP and we're adding CO2 to it, it's going to change into something different, right? But remember, things speed up this process. That's going to speed up the change from changing RUBP into something different by adding carbon dioxide to it. So we're going to use the enzyme, in this case, nope, sorry, it's happened again. I'll get better at these curves. There we go. Okay, we're going to use the enzyme Rubisco. Okay, so really we're starting, I say CO2, we're feeding CO2 into the cycle and it's going to meet up with RUBP, but RUBP is going to join it, be joined to the CO2 by the enzyme Rubisco, right? So because we've now added something to the CO2, it's no longer CO2. So it needs to change into a different substance, right? I'm going to duplicate it again. And what I'm going to do is I'm going to put in here what we get when we put RUBP and CO2 together. under the guide of the enzyme Rubisco, and then what we get is the molecule. Don't fixate on the proper terminology for it. At this point in time, just call it 3PG, right? So, carbon dioxide added to RUBP, catalyzed by the enzyme Rubisco, gives you 3PG, okay? So, 3PG is still not what we want. So we're going to take another step and change 3PG into another compound, right? So again, I'm going to duplicate here and bring it down here. And what we're going to change it from is 3PG. This is a bit confusing, okay? It's a bit confusing, but it's G3P, okay? How I remember the difference between these is if you were to go into your Apple Notes, for example, and you were to type in 3PG, and then you were to type in G3P, and if you were to put it in alphabetical order, the 3PG would go in first. Now, how I remember the way it comes in 3PG is that PG is like the movie ratings you get. You get like a universal, you get a PG, you get 12, 15, 18, and so forth and so on. So, from this, to recap, we're going to go from RUBP, we're going to fix it to carbon dioxide using the enzyme Rubisco, okay? And it's going to catalyze, fix carbon dioxide into 3PG. And now, at the next stage, we need to change 3PG into G3P, okay? Now, how we do that is we need to rely on the substances from the light-dependent stage of photosynthesis, okay, or photolysis. So what two things do we get from that? First of all, we get hydrogen from NADH. Sorry, I've got that wrong. That's NADPH. Remember, it's the photosynthesis, right? So if it's respiration, it's NADH. If it's photosynthesis, it's NADPH. I mean, photo pH right at the end. It should be fairly straightforward for you to remember. The other thing that we're going to get, okay, is ATP. Okay, ATP gives it the energy. It's a cycle. It requires movement energy, so ATP is going to keep everything moving as well as the hydrogen. Right. Going back onto the NADPH, it's going to give hydrogen. Now remember, NADP is what is known as a hydrogen acceptor. A strange way of wording it, but the hydrogen acceptor means that it can accept hydrogen, okay? Think of it like a empty taxi. It's available for passengers, okay? When it becomes loaded, or the taxi becomes full, it then can no longer pick up any more passengers, right? So in this case, the hydrogen acceptor NADP, when it becomes loaded with hydrogen, it then becomes what's known as a reduced hydrogen acceptor, called NADPH, right? Now, when the hydrogen here gets input into the cycle, it means that the NADP is no longer required, right? It doesn't become useless. What it can then do is it can go and pick up more hydrogens from photolysis, keeping the cycle going, right? So essentially what we're doing here is we're adding hydrogen, okay, hydrogen is 3PG and ATP energy, both from photolysis, and we are creating G3P. Right, so the good news is that's the difficult names dealt with at this point in time, okay? So G3P, again, bear in mind it's a series of enzyme steps, is going to have two fates. The first fate, okay, the phosphate is going to be, it's going to be peeled off as glucose, okay? And the second fate is what's left over and not converted to glucose is going to then bring you back to RUBP right here. It makes total sense because you need to get that RUBP again to be able to accept more and more carbon dioxide to keep this cycle turning, to keep producing glucose. If you couldn't produce RUBP, then it stops, okay? It's no good. In terms of high biology, it's no good, right? So the glucose comes off, and the glucose, what you need to know about the glucose, the plant can use it, it can use it immediately for glucose, okay, or it can be used to build up cellulose. So if cell division takes place, plants need to create cellulose for their cell walls in each individual cell, so it can be used for that. And then finally, you've got starch. Okay, starch is like, we've got food cupboards at home, we've got tins of beans that last pretty much forever, okay, as opposed to if you had fresh vegetables, they would only last a week. Tin stuff, save it for a rainy day. It's the same idea. So starch can be used as a storage carbohydrate, like for examples of starch, it might be in seeds, okay, to promote the growth and give them the energy supply for later. So it's like a rainy day saving for that, okay. So that sort of wraps this up. It's not difficult, but there's a few things that you need to be wary of, okay. So from the top, okay, RUBP fixes CO2 via the enzyme Rubisco to create 3PG, okay. So that's pretty much the first step. The second step is hydrogen from photolysis is brought to 3PG, entering the cycle alongside ATP, also from photolysis, and it creates G3P. So we've now got G3P, which can be very, very beneficial to the plant, and it's got two fates. It can either be converted, some can be converted into glucose, which is then going to be used for immediate use by the plant, cellulose, which is going to create new cell walls, or the structure of the new cell walls, and then starch, the storage sugar carbohydrate for a rainy day, so to speak, okay. What's left over and not converted into glucose returns to RUBP to allow it to be fixed again, okay. So another couple of key things. One more thing that you should know about this is it takes place in the stroma, okay. The stroma is like, if you look at a chloroplast, you have got the grana, which are the sort of discs that contain chlorophyll, okay, and the stroma is more like the space, okay. It's the space in between. It's like not the solid green parts containing the photosynthetic pigments, which you got from the earlier on in this, okay. So with that in mind, next week, starting from Monday, I'll go through this again, right. This is just like a practice session to sort of get into it, and just to me check things are working. I'll work on trying to get my writing to be a lot more neater, because I've just conquered how to draw shapes and things on this, but we're getting there, okay. So if you want to join me over the next 3 weeks, Monday to Friday, 9 AM, I'll be doing higher one day, I'll be doing advanced higher the next day, national 5 this day, back to higher human. I'll be doing a lot of things from 9 o'clock in the morning, so you're more than welcome to join. Everything I do will come straight off the SQA arrangement documents, okay. So nothing I'll give you will be me try to impress you with my knowledge. It will just be what you need to know and nothing more, okay. There's absolutely no point of going in a deeper dive into things that are not going to come up in the exam, okay. So if you did join me today, thanks a lot, and drop any questions you might have for me, or even if you want to do questions that you want me to go over, then feel free to drop it in the comments. I'd appreciate if you liked and subscribed to try and get this out there if you feel that I can help you some more. Okay, I'll see you on Monday if anybody's about, or maybe before then. Okay.